Pretty obvious question here. Fungi or other organisms where asexual reproduction is all found. All the options, not everything is related to sexual reproduction. Meiosis is associated with sexual reproduction. So the right choice is what? Meiocyte, a cell which undergoes meiosis. Meiocyte can be either microsporocyte or the megasporocyte. Microsporocyte is the cell which will produce microspores by meiosis. So you can consider that to be a male cell. Megasporocyte is a female cell. But both of them undergoing meiosis to give rise to the gametes, haploid gametes. See, three requirements for sexual reproduction. What are the three requirements? Meiosis should happen. Then gamete formation should happen. And then fertilization should happen. All three should happen. And the sequence is not necessarily meiosis first. Gametes can fuse to form a zygote. Zygote can undergo meiosis to produce haploid spores. That is also possible. If the organism is haploid, see, when meiosis happens for sexual reproduction is dependent on what is the organism's ploidy. If the organism itself is haploid, gametes are produced by mitosis. Fusion of gametes will happen to produce a zygote which is diploid, and then meiosis will give rise to a cell which will undergo further development to become the body. Alright? But all three are necessary. Formation of gametes, fusion of gametes, meiosis, all three. Here conidia are spores again. What type of spores? Conidia are asexual spores. Gemmules are what? Where do you find gemmules? Sponges. You find gemmules in sponges. Gemma cups you find. Sponges these are the asexual reproductive structures. Gemmules are asexual reproductive structures. They are not cells. They are not individual cells. They are structures. Megaspores. Megaspore is produced as a result of meiosis. Generally does not undergo meiosis. It is a product of meiosis generally. Alright. Now megaspore can be a product of mitosis also if the organism is haploid. You understand? Megaspore can be a product of mitosis also when the organism is haploid, and megaspore is a product of meiosis when the organism is diploid. In either case, megaspore will not undergo meiosis. You understand? If the megaspore is a product of mitosis, it will fuse with the microspore, produce a zygote, then the zygote will undergo meiosis. So megaspore is not undergoing meiosis. Meiocyte is the right choice there. Advantage of cleistogamy. Cleisto is what? Closed. Gamy is fertilization. Closed fertilization is cleistogamy. What is the opposite of that? Open fertilization. That means the flower is open. This is in flowers, in plants. The bud does not open and the fertilization happens internally. Internally in flowers is self-fertilization, self-pollination. The pollen is pollinating the same plant, same flowers. What? What is the opposite of cleistogamy? Chasmogamy. Chasmogamy is the opposite of cleistogamy, where the flower opens and it is open for cross pollination. In chasmogamy, in chasmogamous flowers, is it necessary that the pollination is always cross? Is there no possibility of self-pollination in chasmogamy? There is possibility of self-pollination also, but chasmogamous flowers are open for cross-pollination. Cleistogamous flowers are not. Okay, let's see the options. Advantage of cleistogamy is higher genetic variability. Is it so? Self-pollination will reduce genetic variability. More vigorous offspring. Generation after generation, the vigor of the offsprings will decrease. No dependence on pollinators. That is true. No dependence on pollinators is true. Vivipary is what? What is vivipary in plants? Viviparous in animals is? What is viviparous in animals? The offspring develops inside the mother's body. The offspring develops inside the mother's body. So the offspring is born, not maturing inside an egg or something. Vivipary in plant is what? In plants, seeds will start germinating when it is not yet dispersed. While it is still a part of the plant body, the seeds will start germinating. That is vivipary. Okay? Viviparity is not related to cleistogamy. It is related to seed dispersal, delay in the seed dispersal. So advantage of cleistogamy is no dependence on pollinators. Cleistogamy, chasmogamy, you have to consider as opposites. Okay. Monoecious plant of Khara shows the occurrence of antheridium and archegonium on the same plant, stamen and carpel on the same plant, upper antheridium and lower oogonium on the same plant, upper oogonium and lower antheridium on the same plant. It is a matter of remembering it. In Khara, which is monoecious. Monoecious means what? Monoecious is bisexual plant or a homosexual, unisexual plant? Bisexual plant. Bisexual plant is monoecious. Dioecious is unisexual plant. So in a bisexual plant, you will find both antheridium and archegonium. Antheridium giving rise to the anther and archegonium giving rise to the carpel. In this, antheridium and archegonium on the same plant is correct. Which one is upper and which one is lower is something we have to know. In Khara, oogonium, the archegonium is above and the antheridium is below. So right choice is the fourth one, upper oogonium and lower antheridium on the same plant. Got it? You have to remember that perisperm differs from endosperm in origin wise. What is perisperm coming from? Perisperm is originating from nucellus. Nucellus is what ploidy? Assume the parent plant is diploid. Nucellus is diploid. Endosperm is triploid, assuming the parent plant is diploid. If the parent plant is tetraploid, what will be the ploidy of nucellus? If the parent plant is tetraploid, what will be the ploidy of nucellus? Nucellus will be tetraploid. The ploidy of nucellus will be the same as that of the parent plant. The ploidy of endosperm would be hexaploid in this case. If you have to make a general statement, the ploidy of endosperm is 1.5 times the ploidy of the parent plant. 1.5 times the ploidy of the parent plant. That is 2 gametes and 1 endosperm nucleus. Endosperm nucleus is 2 haploid nucleus there, and 1 male gamete fusing with the 2 endosperm nuclei. Is that clear? I am just touching on the key aspects of this. If anybody wants explanation on any of these concepts, repeat explanation, ask for it. Now we will quickly recall the embryo sac. Embryo sac has 2 poles. What are the 2 poles in the embryo sac? Micropylar end, and the other one is callosal end. In the micropylar end, you have 3 cells, haploid cells. Which are the 3 cells? 2 supporting cells, which are synergids, and 1 cell which is egg cell. On the callosal end, you have 3 cells again. All the 3 cells are called antipodal cells. Antipodal cells. In the center, you have 2 nuclei. Those nuclei are called central nuclei. Central nuclei or polar nuclei. After pollination, the pollen tube germinates. The embryo sac is here. Pollen tube is germinating, and it is carrying one male gamete. One male gamete, which will split to produce, which will split by what process? What type of cell division? The male gamete which is coming down will divide by what type of cell division to produce two cells? Mitosis or meiosis? Mitosis. Mitotic division of the male gamete to produce two male gametes. One of the male gametes will go to the micropylar end, break open the synergids, reach the egg cell and fertilize the egg cell. Now this fertilized egg cell will become zygote. Fertilized egg cell will become zygote and develops to produce embryo and cotyledons. Embryo and cotyledons are coming from the zygote which will develop. So what will be the ploidy of all the cells in the embryo and the ploidy of the cotyledon cells? Diploid. Because all of them are coming from the fertilized egg which is the zygote. One more male gamete will come and fuse with the central nuclei. There are already two nuclei, polar nuclei there. Along with that you get one more nucleus. So three haploid nucleus will fuse to form a single nucleus. Now this entire cell, the embryo sac itself is one big cell with multiple cells there. All the other cells will get used up. The zygote will develop to form the embryo and the cotyledons. The endosperm, the fertilized central cell nucleus, the fertilized central cell will develop to form the endosperm. That's all. So in this case here, perisperm is what? The perisperm is around this embryo sac. It is not inside the embryo sac. Outside the embryo sac in the ovule, you have the perisperm. You have the perisperm, which is the nucellus. Nucellus is there. Nucellus will get used up. In most of the seed development, nucellus gets used up. If the nucellus tissue remains, why is it persisting? Why is it continuing to remain? What will be its role later if the nucellus remains as perisperm? Nourishment of the developing embryo. So it is perisperm. Perisperm and endosperm, two things. Perisperm is coming from the nucellus tissue which is outside the embryo sac. Endosperm is the central cell which gets fertilized and develops further to provide nourishment. Both of them provide nourishment. The job of endosperm is also to provide nourishment. The job of perisperm is also to provide nourishment. The ploidy is different. Perisperm differs from endosperm in being a haploid tissue, being a haploid tissue, is it true? No, it is triploid. Having no reserve food, perisperm is diploid. Having no reserve food, wrong. It is for the purpose of having reserve food. Being a diploid tissue, yes. Its formation is by fusion of the secondary nucleus with several sperms, no. So it is diploid. That is the difference between perisperm and endosperm. Endosperm is triploid, perisperm is diploid. Both are reserve food. Endosperm is developed by the fusion of secondary nucleus with one sperm, not several sperms. Which of the following statements is not true of two genes that show 50% recombination frequency? Recombination frequency is an indication of crossing over, genetic variation. Whether the two genes can cross over or not. Whether the two genes can cross over or not. Conversely, recombination frequency also gives you an indication whether there will be independent assortment of the alleles or not. Now here, recombination, can it happen between, crossing over can happen between the corresponding segments of homologous chromosomes, true or false? Crossing over can happen only between the corresponding segments of homologous chromosomes. True or false? Did not understand the statement. Corresponding, see, you consider these three. This is segment 1, segment 2, segment 3. The 3 inches on my index finger. Same on the other index finger also, segment 1, segment 2, segment 3. Now there are 3 segments on both these chromosomes, let us assume. 3 segments on both these chromosomes. Corresponding segment is segment 1 and segment 1 are corresponding segments on these chromosomes. Segment 2 are corresponding segments, which is the same location, same locus. That is the corresponding segment on the homologous chromosomes. Can segment 1 cross over with segment 2, segment 1 of one chromosome cross over with segment 2 of the other homolog? Can that happen? If that happens, what will happen to the alleles? The alleles will change. The locus of the alleles will change. Let us say there is gene 1, gene 2, gene 3 corresponding to the segment 1, segment 2, segment 3. Now if gene 1 on chromosome 1 crosses over with gene 2 of chromosome 2, chromosome 1 will have gene 2, gene 2, gene 3 alleles. There will be 2 alleles of the same gene here. In this, gene 1, gene 1, gene 3. This one will not have gene 2 allele. Is it possible? Cannot this happen? When crossing over is happening, can this not happen? This might happen. If it happens, that particular cell becomes unviable. If that happens, the particular cell becomes unviable. Crossing over, when we are talking about corresponding segment crossovers. So gene 1, there is allele here, there is allele here. It can cross over and exchange the segments. Gene 3 can continue to be so. If gene 1 is crossing over, how likely is gene 2 also to cross over? Here we have gene 1, gene 1, gene 2, gene 3. Same, gene 1, gene 2, gene 3. If gene 1 is having a crossover, synapse, how likely is gene 2 also to have a synapse? Is it more likely or less likely? More likely, is it? Less likely. That is what recombination frequency says. When we are looking at recombination frequency, we are assessing the likelihood of independent assortment. Independent assortment comes into picture when there are more than one genes in question. Correct? So there are 3 genes. Independent assortment of gene 1 and gene 2 versus independent assortment of gene 1 and gene 3. Which combination will have a higher possibility of independent assortment? 1, 2 or 1, 3? 1, 3. Why? They are far from each other. The more farther the genes are from each other, the more are the possibilities of independent assortment. It will certainly happen, cannot be told. But can it happen or not is determined by how close the genes are. If the genes are very close, it is physical contact. So one segment is already in synapse, the other segment gets stretched. There is counterbalance, no? One segment has come closer, the other segment is being pushed away from each other. So for the next segment to come closer, you need to have a certain distance. Recombination frequency is an indication of the closeness of the genes. If the recombination frequency is higher, what does it indicate? The genes are closer to each other or farther from each other. If the recombination frequency is high, distance is more. The distance between the 2 genes is more if the recombination frequency is high. That means those 2 genes are more likely to cross over, independently assort. If the recombination frequency is low, what does that mean? What does that indicate? The genes are closer. The distance is lesser. So with lesser distance and the proximity of the genes being more, recombination will be less. If recombination is less, independent assortment will be lesser. Alright, higher the recombination frequency, higher the independent assortment possibility. Keep this in mind. So recombination frequency of 50% or more means independent assortment is possible. Recombination frequency of 50% or less means they are linked. Linkage is more. Linkage and recombination frequency. How are they related to each other? Directly or inversely? Inversely. If linkage is strong, recombination frequency is low. They cannot cross over. If linkage is weak, recombination frequency is high. So linkage, recombination frequency, independent assortment. Recombination frequency and independent assortment are directly related. Higher the recombination frequency, higher is the possibility of independent assortment. Linkage is inversely related. Put this relationship. Linkage is recombination frequency directly related to independent assortment, inversely related to linkage. All right. Relationship you can write down. Recombination frequency is directly related to independent assortment, is inversely related to linkage. This relationship you make a note. All right. Is this clear? We'll go to the next one. So genes may be on different chromosome. Is it true if recombination frequency is 50%? Genes may be on different chromosomes. True. The genes are tightly linked. Is it true? Genes are not tightly linked here. The genes are loosely linked. Probably, if at all there is linkage. The genes show independent assortment. Yes, if the genes are present on the same chromosome, they undergo more than one crossover in every meiosis. Possible. Statement 2 is the wrong statement with gene frequency 50%. Variation in gene frequencies within populations can occur by chance rather than by natural selection. This is referred to as gene flow, genetic drift, random mating, genetic load. Genetic drift is what? Genetic drift is the right choice there. Genetic flow is what? Movement of alleles between population. Let's say a significant part of one population moves into another population. That is genetic flow. What was not present in the gene pool of this population gets added to the gene pool because of inflow of genes. Inflow of genes into the gene pool by the people coming into the gene pool. There is genetic flow on the other side also, outflow. There is genetic loss there. There is decrease in the gene pool in the place where immigration happened. Where immigration happened, immigration loses genetic diversity. Immigration enriches genetic diversity. All right. Is random mating a contributing factor for genetic drift, or is it a contributing factor for stability of the population, equilibrium state? Stability of a population in terms of its gene pool is maintained by random mating. Non-random mating will lead to genetic drift. Genetic drift is random changes in the allele frequencies. All right. So variation in gene frequencies can happen because of genetic drift. Variation in gene frequencies can happen because of genetic drift, if not for natural selection. What is genetic load? Genetic load. Something happens in a population and it leads to elimination of certain individuals from the population. leading to loss of genes from the population. So this deleterious mutations. Genetic load is the load of abnormal mutations happening on the gene pool. Some mutations happen, deleterious mutations, some damaging mutations happen, and that gets added onto the gene pool, creating a load on the genetic pool of the population. Burden, the load is equivalent to burden. It is a burden. It increases the disease burden. It increases the burden of destructive possibilities on the population. That is genetic load. If two persons with AB blood group marry, two persons with AB blood group note, and have a sufficiently large number of children, these children could be classified as A blood group, AB blood group, B blood group in a 1:2:1 ratio. In a 1:2:1 ratio, modern techniques reveal the presence of both A and B proteins in AB individuals. This is an example of what? See, now there are two individuals with AB blood groups marrying each other. What allele is absent in the in them? What allele is absent in them? I, small I, or IO. The small I allele is absent in them. Meaning what? There is no possibility of having O blood group. O blood group is not possible. So if they have a sufficiently large number of population, large number of children, population of children only you can consider. Large number is a population there. Alright. A, B and B in a 1:2:1 ratio. Modern techniques reveal the presence of both A and B proteins in AB individuals. This is an example of codominance, incomplete dominance, partial dominance, complete dominance. It is codominance. Blood group is an example of codominance, and also blood group is an example of multiple allelism. Multiple allelism. Alright. Partial dominance and complete dominance is not happening there. Both are expressing when they are present. If it is absent, it doesn't express. So there is no incomplete expression. Here, do you recall what is Bombay blood group? What is Bombay blood group? What is the protein which is the precursor to A and B proteins? Which is the primary protein produced by gene I? The blood group gene will produce this protein compulsorily, whether it is IA, IB or small I. There are three alleles for this gene: IA, IB and small I. IA allele will transform this precursor protein into A protein. So there will be precursor protein plus A protein on the RBC. IB allele will make the B protein. So with IB allele, you have the precursor protein plus B protein. Small I allele cannot make either A or B. That means if there is small I allele, only the precursor protein will be present. This is first point. Second point is, with ABO system of blood grouping, absence of an antigen will lead to the individual to develop antibodies against the antigen. If A is absent, A antigen is absent, A protein is absent, that is A antigen is absent, the person's blood is capable of producing antibody against A. For all other antigen-antibody reactions, there should be exposure to an antigen before the body develops active immunity against that antigen. But ABO, there is no need for exposure to the antigen. Absence of the antigen by default will make the body develop active immunity against that antigen. So if A protein is absent, the person can produce antibody against A. So all the blood groups without A protein will have the individuals with any other blood group without A protein can produce antibody against A. So will persons with B blood group produce antibody against A? The person's blood grouping on a regular bedside test will show to be O positive or O negative. O is the blood group that will show up. But a Bombay blood group person, a normal O blood group person will be producing H antigen, precursor antigen. But a Bombay blood group person will not be producing H antigen. So his body is capable of producing antibody against H antigen. When you test, you find the person to be of O blood group because no A, no B is O. You transfuse O blood group blood into the person. He is capable of producing antibody against H antigen. The normal O group person's blood has H antigen. Antigen-antibody reaction will happen. Alright. Remember H antigen. That is the precursor antigen. Bombay blood group is defect in the gene which leads to no production of H antigen. Is that clear? We should not be repeating this. With every revision, this should become very, very clear in your head. Any question related to Bombay blood group will be a higher level question. You should have advantage with that. And blood groups, remember, no antigen, antibody by default. No antigen, antibody by default. When we are doing blood transfusion, antigen of the donor is important. Antigen of the donor is important, not the antibody of the donor. Because antibody production is dynamic. It can increase depending on the antigen load. But antibody present in the donor's blood may not cause a serious reaction on the antigen present on the recipient's RBC. Alright. No antigen, default antibody production, only for ABO system, not for D, not for D antigen. D antigen is related to which blood group system? RH. RH blood group is D antigen. So what we say positive, negative for blood groups is D antigen. Absence of D antigen, there is no by default antibody production. Exposure will trigger the body to produce antibodies. You understand? If D antigen is absent in your blood, you are not producing antibody by default. Only when you are exposed to D antigen, your body will learn to produce antibody. ABO system, no antigen, antibody by default. H antigen is the precursor antigen. Even O blood group people will have H antigen. Absence of H antigen, Bombay blood group. Absence of H antigen, Bombay blood group. What blood can be given to Bombay blood group person? Can we give O blood group to Bombay blood group person? No, because O blood group will have H antigen. The donor for a Bombay blood group person should be another Bombay blood group person only, not anybody else. Understand? This much you should remember with blood groups. The process by which organisms with different evolutionary histories evolve similar phenotypic adaptations in response to a common environmental challenge is called evolutionarily different, similar characters. That is, what type of evolution? Convergent evolution. Convergent evolution. People from organisms evolving in different conditions, in different gene lines, developing in similar conditions, develop similar characters. Convergent evolution. What are the types of organs that develop in convergent evolution? What organs develop? Convergent evolution leads to the development of what organs? Analogous organs. Analogous organs. Different structure, similar function. Example, wings of bat and wings of butterfly. Both are used for flying, but the origin, the developmental origin is different. Convergent evolution. Opposite is similar development, different functions. Divergent evolution, organs are homologous organs. Example? Flippers of whale and wings of bird. Both are developing from the similar structure only. Both are developing from the similar structure, but because of the conditions in which they have evolved, their functions are different. Structurally same or evolutionarily same, but functionally different. Structurally same, functionally different, homologous organs. Structurally different, functionally same, analogous organs. Homology is a characteristic of divergent evolution. Analogy is a characteristic of convergent evolution. Adaptive radiation is what? Adaptive radiation. Can we say adaptive radiation is also divergent evolution? Radiation is to move from a common point to different destinations. Source is common, destination is different. That is radiation, no? Adaptive radiation is the adaptations leading to different appearances. Polar bear and a sloth bear, are they same? Are both bears? They are both bears. Structurally, physically, everything they are same. But their body characteristics have changed. Though both of them are bears, they appear different. Morphological and phenotypical characteristics vary. Those variations are a result of adaptive radiation. Human beings in different regions have different characteristics, different appearances phenotypically. Can we not say it is adaptive radiation? People living in tropics have more pigmentation than people living in temperate zones. That is also adaptive radiation only. Evolutionarily, we all belong to the same source. But because of our adaptations in different conditions, we have developed different characteristics. That is adaptive radiation. Diversification of a species into multiple forms adapted to different environments. That is adaptive radiation. The tendency of a population to remain in genetic equilibrium may be disturbed by. What will contribute to genetic equilibrium, you tell me first. What do we call that equilibrium? What is the equilibrium we are talking about here? If a population is in genetic equilibrium, we call that equilibrium by a name. What equilibrium is that? Hardy-Weinberg equilibrium. What are the conditions which will contribute to Hardy-Weinberg equilibrium? Population should be large. It should be a large population. Mating should be random. No migration, whether it is immigration or immigration, both. There is no migration. Correct? Those are all conditions which will lead to, and random mating. Random mating should be there. These are all conditions which will contribute to the population remaining in a state of equilibrium, genetic equilibrium. Alright, what contributes? What can be disturbed? What can disturb the genetic equilibrium? Random mating. Is it pro-equilibrium or anti-equilibrium? Random mating. Pro-equilibrium. Lack of migration. Pro-equilibrium. Disturbances will be less. Lack of mutations. Pro-equilibrium. Mutations will bring in variations that will disturb the equilibrium. Lack of random mating. Anti-equilibrium. So, tendency of a population to remain in genetic equilibrium can be disturbed by lack of random mating. Presence of random mating contributes to equilibrium. Now, we will recall Hardy-Weinberg equilibrium. There is gene frequency and there is genotype frequency. And here we are assuming every gene to have 2 alleles. We are assuming every gene to have 2 alleles, and one of the allele is dominant, the other one is recessive. So Hardy-Weinberg equilibrium is conveniently eliminating multiple allelism, polygenic inheritance, and codominance, partial dominance. All of those variations are removed. That means Hardy-Weinberg equilibrium is an illusion. It is just a theoretical exercise. There cannot be genetic equilibrium. Theoretically, that is the ideal state. It remains in equilibrium. So what do you represent? How do you represent the genetic equilibrium? Give me the equation for genetic equilibrium. Frequencies are represented with reference to 1. Frequency is always represented with reference to 1, not as a percentage, as a proportion of unity. P plus Q is equal to 1. P is what? P and Q here, are they allelic frequencies or are they genotypic frequencies? Allele frequency. P refers to frequency of dominant allele. Q refers to frequency of recessive allele. P square plus 2PQ plus Q square is equal to 1. P square refers to homozygous dominant. 2PQ refers to refers to heterozygous condition, and Q square refers to homozygous recessive condition. So these two equations you have to remember. P plus Q is equal to 1, P square plus 2PQ plus Q square is equal to 1. That's all. Whatever is given, we can calculate the others. Let us say in a population of 100 individuals, 25 individuals represent homozygous recessive state. What is the frequency of the dominant allele? You tell me now. Frequency of the dominant allele. Calculate and tell me. In a population of 100 individuals, the frequency of the dominant, the frequency of the recessive allele is 25. 25 individuals are homozygous recessive for a particular gene. What is the frequency of the dominant allele?
0.5.
What is given? See, when we are solving Hardy-Weinberg equation based problems, total number of individuals, how many? 100. What is given? Number of homozygous recessive individuals. So in the equation, what is given? Q square. Q square is 25 out of 100. So to bring it down to 1, you have to always write it as 25 out of 100 divided by 100. So Q square is equal to 25 by 100, that is equal to 1 by 4. Q is equal to square root of 1 by 4, which is 1 by 2, which is 0.5. So Q is 0.5. The frequency of the recessive allele is 0.5. What do we have to find out? The frequency of the dominant allele, which is P. When you know Q, which is 0.5, P plus Q is equal to 1, P should be 0.5. Alright. In a population of 100 butterflies, in a particular variety of butterflies, brown wings are dominant and red wings are recessive. Brown wings are dominant and red wings are recessive. If the red-winged butterflies in a population of 100 butterflies are 36, how many butterflies are homozygous for brown wing? How many butterflies are homozygous for brown wing? Brown wing is dominant, red wing is recessive. In a population of 100 butterflies, if 36 are red-winged, how many are homozygous for brown wing? Okay. Again, total number 100, dominant, brown, recessive, red-winged individuals 36 out of 100. 36 out of 100. 36 out of 100. That is 9 out of 10. 9 out of 25. 9 by 25. Q square is 9 by 25. What do we have to find out? P square. How many are homozygous for brown wing? So we have to find out Q square. P square is 36 divided by 100. So Q is square root of 36 divided by 100. That is square root of 36 by 100 is 6 by 10. 6 divided by 10, that is 0.6. Q is 0.6. By that you know what is P. 0.4. P square is 0.16. 0.16 out of 1. How many out of 100? You'll have to multiply into 100. 16. 16 butterflies will be homozygous for brown wing. It is very simple calculation, but you have to always do it in a sequence. 100 butterflies total. Dominant brown, with that is P. Recessive red, that is Q. Red ones 36 out of 100, that is Q square. Correct? So Q square is equal to 36 divided by 100, which is Q is equal to square root of 36 by 100, that is 6 by 10, that is 0.6. If Q is equal to 0.6, P is equal to 0.4. Because P plus Q is equal to 1. Correct? Now what is asked? Homozygous for brown is the question. That is P square is what is being asked. Homozygous for brown, which is the dominant one, is the question. So P square is being asked. We found out P is 0.4. So P square is 0.4 into 0.4. That is 6.16. P square is 0.4 into 0.4. That is 0.16 out of 1 is equal to 16 out of 100. Out of 100, it is totally 16. Calculation part is easy. Logic is what you have to get here. Hardy-Weinberg equilibrium. Very likely question. Hardy-Weinberg equilibrium is like a stock question. So to make it a little bit fun, just add some Hardy-Weinberg equilibrium calculations. If you want to make it more fun, make the question long. Give stories. Okay. You should always pick up this. You should always look for what is the total number, what is it, what is given, other details, what is asked. That's all. What have they given? What are they asking for? What is the total number? Gather all the data. Translate the data into equation. So ultimately you have to find out what is P and Q. If you find out what is P and Q, you can find out everything else. So based on what is given, you figure out. Did everybody understand this? Clear? Let's move forward. There should be no problem there. Once you get the hang of it, you can just quickly do it in your head. Okay, I'll give you one more. I'm trying to make it complex. Okay. In the same example, brown and red winged butterflies, if 42 butterflies are, if 42 butterflies are, if 42 butterflies are heterozygous for brown color, if 42 butterflies are heterozygous for brown colored wing, if 42 butterflies are heterozygous for brown colored wing, and the frequency of recessive allele is 0.3, and the frequency of the frequency of the allele, the frequency of the recessive allele is 0.3. How many are homozygous for? How many are homozygous for brown wing?
49.
Okay. If 42 butterflies are heterozygous for brown colored wing and the allele frequency of recessive allele is 0.3, how many butterflies are homozygous for brown colored wing? See, in this, heterozygous is a distraction. Total number is 100. Frequency of recessive allele is 0.3, meaning frequency of dominant allele is 0.7. How many are homozygous for brown wing? What do you have to find out? What do you have to find out? P square. You have to find out P square. 0.7 is P. P square is 0.49. 7 sevens are 49. 0.49 out of 1. And you have 100 individuals there. 100 are there, so 49. 49 out of 100. 49, 42, 9. Got it? Did not understand? What information have I given? Total number is keep it on a side. Total number 100. What is the information? What other information did I give you? 42 are heterozygous. 42 are heterozygous. So 2PQ is 42. And I also told the frequency of recessive allele is 0.3. What did I ask for? How many individuals are homozygous for brown colored wing? How many individuals are homozygous for brown colored wing? So brown colored wing is dominant. What are we supposed to find out? P square is question mark. That's all. This information is not required only. To find out P square, what do we have to find first? P. So the solution, I'm solving like mathematics of 6th standard. The solution is P plus Q is equal to 1 given Q is equal to 0.3, substituting Q in the equation, substituting for Q in the equation. Alright, that is how we do now. Substituting for Q in the equation, I'll just write it as implies P plus 0.3 is equal to 1. P is equal to, I'll shift it this side. So 1 minus 0.3. So P is equal to 0.7. Now, if P is equal to 0.7, P square is equal to 0.7 into 0.7. So P square is equal to 0.49. Okay? 0.49 is for 1. How much is for 100? Cross multiplication. Cross multiplication 0.49 to 1 x to 100 x into 1 is equal to 0.49 into 100. This is cross multiplication. X is equal to 49. That's all. So P square basically. Okay. 49. X is the total number of individuals who are homozygous dominant in the population. Total number will be given such that it will be easy for you to calculate. Because in a biology, in the NEET paper, the objective is not to check your calculation skills. The objective is to check your logic. Is this clear? Don't do mistakes on Hardy Weinberg. Very easy. Give you marks. Let's move on. Which of the following BT crops is being grown in India by the farmers? What is BT crops? Is it biotechnology crops? Biotechnology crops also, but BT does not refer to biotechnology. BT refers to Bacillus thuringiensis. Bacillus thuringiensis crops are unique in a way. See, is Bacillus thuringiensis BT crops good for the consumer or good for the farmer? Is BT crop good for the consumer or good for the farmer? Why? You can say for both. Why is it good for the farmer? But what is BT crop in the first place? There is a protein called cry protein, cry gene, crystalline protein gene, which is like a blade, for insect gut. Insects which eat the cry protein containing plant products, maybe here it is cotton or BT brinjal, corn. When they eat it, the cry protein, when it goes into the insect gut, it will cut the insect gut. It doesn't get digested. It will act as a blade on the insect gut, cutting the insect gut, leading to the insect getting killed. The insect will bleed internally and it will die. So the insects also learn. The insects learn to not eat those crops. They learn not to eat those crops. It is behavioral training for the insects. Some of these fellows go and feed on a particular farmland. Those who feed on that farmland die more frequently than those who feed on some other farmland. So the research team of the insects will tell them, there is some problem here, don't eat from that. Right. Otherwise also it's like a natural. Now the problem is, cry protein is small. Cry protein is small, the insect is also small. It is cutting the insect's gut and damaging the insect's gut. Killing the insect in a short span. How do we know that the protein is not doing the same thing to our gut also? Because our gut is big. Our gut is big compared to the insect's gut, and the damage will not be significant enough to kill us or cause illness in us. Can we rule out the possibility of these cry proteins damaging our gut as well? That is the ethical consideration, which is blocking the introduction of Bt gene or the Bacillus thuringiensis cry protein gene into multiple other crops. In India, the permission has been given for cotton. I think in 2002 it was given. Bt cotton is being grown. Why cotton has been allowed? Why is it allowed to cultivate Bt cotton and not Bt brinjal or Bt corn? We don't eat cotton. We don't eat cotton fibers. Cotton is used for yarn, clothes. So it is allowed. Bt brinjal, there was a big debate on bringing in Bt brinjal, but Bt brinjal has been refused. We are not allowing Bt brinjal to be grown. India takes a skeptical stand on Bt crops, especially when we are cultivating for consumption. Bt brinjal is, I think, permitted in some countries. Bangladesh probably has Bt brinjal permission. Like that. So this is Bt. What is it against in cotton? Bt cotton is against what insect? Bollworm. Bollworm. Remember that. Bt cotton is bollworm is the common insect against cotton. So Bt cotton is cultivated where there is bollworm in pesticide pest. Okay. So cotton is being grown in India by farmers. Crystalline protein, cry protein, you should know. I am of the personal opinion that it will damage human gut also. And why do I say it is beneficial for consumers as well? Farmers will use something or the other to ensure that their produce is protected from insects and pests, and they have a higher yield. They have a higher yield. For a farmer, yield is of importance. Yield is of significance. That is why what they do, or many of them do, load with pesticides and insecticides. Pesticides and insecticides are not surface chemicals. When you spray insecticides and pesticides onto the crops will absorb these chemicals. So pesticide sprayed foods, let's say fruits and vegetables, how much ever you wash the surface of the fruit and vegetable, you will not be removing the pesticide from that completely or insecticide from that completely. It would have gone into the cells of this produce. How will you remove that? When you consume it, you are also consuming the pesticides and insecticides. So we are constantly eating poison along with food. And these pesticides and insecticides are constant cellular irritants. They can irritate the cells. When this irritation happens continuously for a longer period of time, in a person who has susceptibility, cells can turn into cancers. Why are we seeing more incidents of cancer? One, we are surrounding ourselves with a lot of radiation. Radiation also is constantly irritating the cells. Constant irritation of the cell is a cause of cancerous modification of the cell. It is metabolic breakdown only, no? If the cell loses the ability of contact inhibition, it is metabolic breakdown. It has lost a particular ability. So something has to trigger that. It cannot suddenly spontaneously get destroyed. Something has to trigger it. And what is triggering it? Constant exposure to radiation and constant exposure to chemicals. Chemically induced and radiologically induced metabolic breakdown is, in my opinion, a significant contributing factor for cancers. We are seeing increase in cancers. So treatment of cancer, on one part, treatment of cancer is by developing more and more anti-cancer drugs. But prevention of cancer is by lifestyle modification only. We have to modify the lifestyle. At least food, we have to eat proper food. We should not keep loading our body with chemicals. Food. Everything comes down to food in my advice. Alright. So, I have set up a residential campus in Bangalore also now. only for NEET training, exclusively NEET, no training for engineering entrance or anything. Completely residential, 2 year, full 2 years, and with the guarantee of a government medical college seat. This same process, what we are doing here, it will be more intense. The principles and processes what we are applying is the same. This is a blueprint. What we are training you on, what I am training you on is a blueprint for success. Eventually, see sir, even after your Star 30 program, once you get into MBBS, even then you can continue to be guided on the success blueprint. I have a community where I guide students on implementing the success blueprint. I invite all of you to be a part of that community and continue to be associated. So you learn and practice implementation of the success blueprint. This is the beginning of your journey, not the end of your journey. So somehow you might scrape through, let's assume, you might scrape through and get into a medical college. May not be a great medical college, an average medical college, whatever it is, you get in and you do something. But the journey continues. If you continue to practice what you have been made to practice in the last 6 months, this is a very concentrated program. Not giving enough time for every stage to evolve in you because of the constraint of time. But if you continue to practice that for your MD or MS, for your post-graduation, you can aspire to be in AIIMS or such similar top-class medical institutions. For further education, move further. It's always an upward movement. And it is all self-driven. You don't need external support. You just need support in mastering the blueprint. Whatever blueprint I am training you on right now, for few more years you have to practice it. You have to master it. Okay. So in our residential campus, same, 1 hour of yoga every day. Here it is difficult to have yoga trainers to involve and take you through stages of development. So 2 years for mastering yoga also. Complete control over body mind. Every day 1 hour of play, same, one cinema every week. All of that remains the same. Food wise, it will be a completely nutritious, balanced vegetarian meal. No sugar, no maida and no deep fried items for 2 years, imagine. No sugar at all, you will forget what sugar is for 2 years. So chocolates, biscuits, all of that eliminated. Bread and bun and biscuits are also eliminated because maida, refined flour, that is bad on gut. See, food is so important. Your gut determines how intelligent you will become. Your intestinal bacteria will control your intelligence. So you keep the intestinal bacteria healthy, you keep a good healthy intestinal environment, your brain functioning becomes better. It is so complexly designed and it is so intricate. The beauty of it is you don't have to know it. You just have to do what is right. Do what is right, automatically everything will fall into place. Alright, anyways. BT. So BT produce hybrid foods, all the insecticide, pesticide laden foods we need to avoid, but unfortunately that is possible only if we grow our own food. As long as we are buying food from the market, we are not sure of that. Anyways, let's cut that short. A good producer of citric acid. What produces citric acid? In industrial production, what do you use? Which bacteria do you use for citric acid production industrially? Aspergillus niger. Aspergillus niger is used for industrial production of citric acid. Pseudomonas, Clostridium, nitrogen fixers. Pseudomonas and Clostridium are nitrogen fixers. Pseudomonas aeruginosa, Clostridium. Clostridium is also, you have to remember, for disease. Clostridium tetani, Clostridium botulinum, and there is one more Clostridium. One more species of Clostridium causes gas gangrene. Clostridium tetani causes tetanus. Clostridium botulinum is a very powerful toxin. Produces a very powerful toxin. It's a toxin and it can kill instantly. Botulinum toxin, neurotoxin, blood toxin and neurotoxin. Then Clostridium, one more Clostridium causes gas gangrene. 3 deadly diseases are caused by Clostridium: tetanus, gas gangrene, and botulism. One is a food poison, botulinum toxin. So Saccharomyces is used for Saccharomyces cerevisiae, ethanol production. Aspergillus niger and Saccharomyces, both you have to remember for industrial production of citric acid and ethanol respectively. Next, DNA fragments generated by the restriction endonucleases in a chemical reaction can be separated by centrifugation. Is used for what? There are 4 processes given. We look at what each one is used for. What do you use centrifugation for? What does a centrifuge do? Separates substances based on its densities. Centrifuge is used for separating substances based on densities. Alright? Continuously rotate and remove separate substances based on densities. Mixture of substances, you centrifuge it, they will all separate based on their densities, and you can use whichever fragment you want. Electrophoresis, electrical charge applied on components which can be separated based on their charge. Do we use electrophoresis for DNA fragment separation? Yes. What is the DNA is what charge, net charge on DNA is? Positive or negative charge? Negatively charged. DNA is negatively charged. Okay. So what is the medium that you use for electrophoresis? Agarose gel medium. Already. PCR is for what? Amplification of DNA fragments, polymerase chain reaction, not used for separation. You use PCR to amplify selected DNA fragments. What is a special enzyme that is used in PCR? Taq polymerase. Taq polymerase is coming from which organism? Thermus aquaticus. What is it? What is Thermus aquaticus? What is it by classification? What is it? It is a bacteria. What type of bacteria? Archaebacteria. Archaebacteria which can withstand, the Taq polymerase enzyme can withstand very high temperatures, which is used in PCR, polymerase chain reaction. Alright. Restriction mapping is? What is restriction mapping? Restriction mapping is used to identify fragments based on the restriction sites, not for separation of fragments. You know which restriction enzyme can act at which place on the DNA. What is the sequence of nucleotides which the restriction enzymes act on? What do you call that? Palindromic sequences. Palindromic sequences. Restriction endonuclease classification, do you remember? 1, 2, 3, 4. Which class of restriction endonucleases are used for genetic engineering? 2. Restriction endonuclease 1 produces blunt ends. Restriction endonuclease 3 and 4 produces methylation. Restriction endonuclease 2 doesn't methylate the ends and produces sticky ends. Restriction endonuclease 2. Anyways. Which of the following is not correctly matched for the organism and its cell wall degrading enzyme? That means which organism's cell wall can be degraded by what enzyme? Bacteria, lysozyme. Can lysozyme enzyme degrade the bacterial cell wall? Yes. Which secretion in your body produces lysozyme? In which secretion is there lysozyme in your body? This secretion is associated with protecting something which is not having, which is exposed to the surroundings. Tears. Tear gland secretion. Tears has lysozyme. See, so beautifully designed. Tears coat your conjunctiva, the sclera, exposed to the exterior. So because it is moist. If it is not moist, it will lead to frictional damage of the conjunctiva. So you have to keep it moist. Moisture is a favorable environment for bacterial cultivation, bacterial growth. It is exposed to the surroundings. Moisture is there. There is bacteria all around us. It can go and settle down and grow there. But unless sometimes now and then when our eyes get infected, otherwise mostly our eyes are clear. It is not getting infected by bacteria which is all around us and constantly coming in contact with the eyeball. Lysozyme is protecting it. So if your eyes become dry eyes can lead to more infection. There is moisture and warmth anyways, but if the dryness increases, if the moisture becomes lesser, you are more likely to get infected because of friction also. Friction causes micro injuries. Those injuries offer conducive environment for the bacteria to grow. So tears is so important. Alright. Plant cell wall is made up of cellulose. Cellulase is used for breaking down cellulose. There are 3 layers in the cell wall. Primary cell wall is made up of cellulose. Secondary cell wall is made up of lignin. Middle lamella is made up of pectin, calcium pectinate, pectin. So pectinase is used for separating the plant cells, breaking them, detaching them from each other, pectinase. Cellulase is used for breaking down, completely destroying the cell. And pectin, calcium pectinate, pectin is responsible for turbidity in fruit juices. Pectin is responsible for turbidity in fruit juices. To clarify fruit juices, if you want to make it clear, you add pectinase enzyme which will degrade the pectin and convert that into sugars also. So the sweetness of fruit juices increase by adding pectinase and it also becomes more clear. Alright, you make apple juice, you add pectinase and carbonate it, it becomes appy. Alright, natural appy. If you have a cylinder of carbon dioxide, soda maker, you have soda maker, and if you can get access to pectinase enzyme, I have tried to get pectinase enzyme. I like making juices, experimenting with juices. In the early days of my experimentation when I was in school, we were in a rented house in our village. The owner of the house came one day. I was experimenting with some juice. I don't know what all I added into that juice. Unfortunately for him, he came at the same time when I was about to finish the juice. With a lot of excitement, I gave it to him. Uncle, please drink this juice. I made a new juice. He drank it. Without a word, he just left. I don't know what was the purpose that he had come home for. But by the time I took the glass from his hand and kept it in the sink and came back, he was not there. He had just left. Maybe he had developed nausea and he wanted to vomit and he did not want to do it. Make me feel sad. I have not got pectinase enzyme. It is not available in retail. Maybe it is available only for industries. I am still continuing to figure out where I can get pectinase enzyme so that I can make appy at home. Make appy at home, drink it and be happy. You don't even have to add sugar. Apple juice itself is sweet. When you break down pectin by pectinase enzyme, it will make it even more sweeter. So without sugar, you will have a sweet apple juice. Carbonate it by using a soda maker. It becomes fizzy apple juice, appy fizz. Okay. We'll do 2 more, 1:30 till 1:30 we will do, and we'll take a 5-minute break, not a 10-minute break. Only for those who want to go to the washroom. Otherwise, sit here and sleep for 5 minutes. Don't go out and do your conference meetings, mini conferences. Alright, next one, 129. Okay, plant cell cellulase, algae methylase, is it right? Methylase is what? Methylation. Methylase brings about methylation of DNA. It's not a cell wall degrading enzyme. Fungi, chitinase. Fungi have chitin in their cell wall, so that is also true. Option 3 is wrong. Colonies of recombinant bacteria. Oh, this one is a good question. The colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because of. See here, there is no mention of what bacteria it is. Only mention is recombinant bacteria show white color colonies, Non-recombinant bacteria show blue color colonies. So we have to look at the options and figure out what bacteria this might be and what is the possibility. This is a good question. And this is based on what we have already studied, lactoperon, lactose related thing, and E. coli is probably the bacterium. Non-recombinant bacteria containing beta-galactosidase, possible, because in non-recombinant bacteria, beta-galactosidase will lead to the breakdown of lactose into glucose and galactose, leading to the blue coloration. Blue coloration pigmentation is because of the presence of galactose when the lactose is broken down. Insertional inactivation of alpha-galactosidase in non-recombinant bacteria. Now, we have seen lactoperon with 3 genes and 3 enzymes. ZYA. What are the enzymes? LAC-Z produces beta-galactosidase. Y produces lactose permease. And A produces trans-acetylase. So, insertion option is given as alpha-galactosidase. Let us see, wait. 3. Insertional inactivation of alpha-galactosidase in recombinant bacteria. So, option 2 and 3 are talking about insertional inactivation of a particular enzyme. One in non-recombinant and one in recombinant. If at all insertional inactivation is to be considered, we should consider that in non-recombinant bacteria or recombinant bacteria. Recombinant bacteria, because you are inserting the gene of interest in the site of a existing gene. So the existing gene has been inactivated. Insertional inactivation is that. Option 2 is eliminated because non-recombinants are the ones which do not have the gene of interest. If you are not inserting the gene of interest, where is the question of a particular gene of the bacteria getting inactivated? So insertional inactivation is seen only in recombinants, not in non-recombinants. Alright. 4. Inactivation of glycosidase enzyme in recombinant bacteria. There is no glycosidase enzyme, there is galactosidase enzyme here. So option 4 is wrong in its statement only. So it has to be either 1 or 3. It has to be either 1 or 3. Non-recombinant bacteria contains beta-galactosidase. Possible. Now, insertional inactivation of alpha-galactosidase in recombinant bacteria. Do we have alpha-galactosidase? Now here we should assume that it is Escherichia coli, E. coli. Alpha-galactosidase is also present. Alpha-galactosidase is also present in another operon. There is another operon, MEL-A operon, where alpha-galactosidase is present. Alright, Melibbiose is also a disaccharide. Melibbiose is also another sugar. There is an operon which operates for breaking down of Melibbiose. That is the MEL-A operon, where you find alpha-galactosidase also. So alpha-galactosidase inactivation and beta-galactosidase inactivation, both are possible. Statement 1 is talking about beta-galactosidase being present in non-recombinants, but it is not talking about whether it is present or absent in recombinants. So between 1 and 3, what should be considered as a better option? 3 is a better option because it is talking about insertional inactivation of a relevant enzyme and recombinants. Because lactoperon is not specifically mentioned, we should consider option 3. This one appears like a simple question, but I personally feel this is an out of context question, though not out of syllabus. Out of context. Because you have not learnt lac MEL-A operon. You have learnt only the lactoperon. Alright, anyways. Last one before we take a break. Which of the following are likely to be present in deep sea water? Now, deep sea conditions, you tell me, what might be a problem in deep sea? Light penetration is not available. If light penetration does not happen, what cannot be present? photosynthetic organisms cannot be present in deep sea. Correct? High pressure, deep sea, high pressure. Oxygen availability also will be lesser. Oxygen light availability will be lesser and high pressure. So who can sustain in deep sea is what we have to see. Archaebacteria, eubacteria, blue-green algae, saprophytic fungi. Now saprophytic fungi are mostly terrestrial. You don't find saprophytic fungi growing in deep sea. They are in moist and warm areas. Saprophytic fungi are mostly terrestrial, so rule out. Blue-green algae are producers. Blue-green algae are producers, therefore they will also not be able to survive in deep sea because there is no light penetration. Between Archaebacteria and eubacteria, which one has a higher likelihood of being present in deep sea? The question is not talking about which may not be present at all. Eubacteria also might be present. Many other organisms might be present. But the likelihood of existing in deep sea is higher for Archaebacteria. So in questions, you should always notice whether something is an absolute assertion. When there is an absolute assertion, then there cannot be exceptions. What is a range, likelihood, possibility, wordings you have to pay attention. You have to pay attention to the wordings. Look at the question. Which of the following are likely to be present in deep sea water? It is neither talking about present only in deep sea water or certainly present in deep sea water. No. Likelihood. When you are looking at likelihood, you should analyze the survivability. Who is more likely to be present in Star 30 campus? Option A, those who have the willpower to stay without mobile phones. Option B, those who are very attached to their parents. Option C, those who love, sleeping on the bed and studying. Option D, those who enjoy studying while eating and listening to music. Who are more likely to survive in Star 30 program? Option A. No guarantee that they will also survive. Alright, some of you wanted to go home, no? Run away. Running master. For whatever reasons. But likelihood is those who have the willpower to stay away from mobile phones can survive. Alright. Others cannot survive. Those who already have a relationship to maintain, they will start having insecurities of losing the relationship in 1 year. What if I am not in touch? Such people cannot survive. People who can just cut off from everything else and stay happily here can survive. Alright. So in our Bangalore residential campus, who can survive? Those who can stay like that for 2 years. No phone. The same, parents will call 1 week and visit 1 week. That children will be more than happy. Especially in Bangalore. Children will be more than happy if parents don't have access to them. Alright. Because parents are constant motivators and demotivators every day morning when they are waking up. Get up. If you don't wake up early, gone. You are waking up at 7 o'clock. If you are like this, nothing will happen to your life. You are useless. Otherwise, hyper motivation. Come on, you have to do it. You can do it. Let me do it. Please stop. Don't give me motivation on a daily basis. Alright? Okay. We'll take a short break. 5 minutes. 11:10 now, 11:15. Everybody should be back. Phosphorus. Phosphorus is present in seawater, animal bones, rocks, as well as fossils. Alright. What is the predominant component in seawater? Sodium. Sodium and chloride. Phosphorus is present mainly in salt, rock. Rock. See, there are two types of salt that you get in the market. One is sea salt, what the regular table salt that we use. The other one is rock salt. Rock salt is more expensive. Why rock salt is more expensive? It is rich in potassium, not so easily available as sea salt, and it is not so easy to process rock salt and bring it onto your table. More healthy also because rock salt gives you the same saltiness. It gives you the taste for food, but it is low in sodium and high in potassium. Low sodium content of the rock salt is good for hypertensive people, cardiac patients, hypertensive patients, low sodium diet requiring patients. All of them will benefit from rock salt. Rocks are the reservoir of potassium. Bones also are the reservoir. Bones also store potassium, but the proportion of potassium found in rocks is much higher than what is found in bones. Seawater also contains potassium, but you cannot consider that as a reservoir of potassium. Fossils also have it, but small amounts. Secondary productivity is the rate of formation of new organic matter by producer, parasite, consumer, decomposer. Productivity is the addition of biomass. How much biomass is being added by a particular organism is called productivity. By a particular trophic level, you can say, in a particular region. Primary productivity is what is produced by producers. Primary productivity is of the producers. Consumers consume the producers, and then they also transform the energy or the mass into productivity in their body. They also add on to the biomass in their body. That is secondary productivity. In all these productivity, you have gross productivity and net productivity. Gross primary productivity, net primary productivity, gross secondary productivity and net secondary productivity. Gross productivity is biomass generated before loss. Before loss in terms of consumption, that is utilization for their own respiratory needs, as well as loss in the form of excretion. Alright. So productivity is what gets established, what gets stabilized in the body as biomass. That is productivity. And you look at it for the entire trophic level. So primary productivity is of producers, secondary productivity is of consumers. Decomposers we should not consider for productivity. They are the ones who will degrade, they will break down. Should we consider parasites for productivity? Should we consider parasites for productivity? They also are accumulating biomass, no? They are taking material from your body and they are also converting that into mass in their body, biomass in their body. Generally, parasites are also not considered as a part of calculating productivity. Secondary productivity is specifically reference to consumers and not parasites or decomposers. Right? Which one of the following is not used for ex situ plant conservation? In situ and ex situ. In situ means what? In their natural habitat. Ex situ is outside of their natural habitat. So which of the following is considered, we will see. Then we will look at what is not used. Field gene banks. Are they used for in situ conservation or ex situ conservation? Are field gene banks places of conservation of plant? They are. What is a field gene bank? That is a new form of dance, is it? Okay. No, only 2 people understood. Or some 4 or 5 people around the dancers would understand. Others don't understand. Anyways. Field gene banks are the places where you are cultivating specific plants to protect them. Alright. Seed banks, are they conservation methods? Are they places of conservation? They are also places of conservation of plants where you are storing seeds of certain plants. Botanical gardens are also ex situ conservation techniques. Shifting cultivation is not conservation technique. It is utilizing different pieces of land, different parcels of land for cultivating in a cyclical manner. It is a cultivation practice and not a conservation method. So shifting cultivation is not used for ex situ plant conservation. Kyoto Protocol was endorsed at COP3, COP5, COP6, COP4. What is COP? For any agreement, these are all multilateral or multinational agreements. That means many countries are involved. Many countries are involved in coming into an agreement where they will agree on certain deliverables, certain actionables related to a specific challenge. Kyoto Protocol relates to certain agreements that the countries that participated in this conference agreed upon to act for overcoming the challenge of pollution, environmental pollution. COP is Conference of Parties. That's all. Who are all the parties? Parties are all those countries which participated in the conference. Conference of Parties 3. There are multiple conferences that happen for climate change management. COP3 happened in 1997 in Kyoto, Japan. Kyoto Protocol was given then. So what is the general agreement? See, Kyoto Protocol or any other such environmental agreements that come into play are all part of United Nations Framework Convention on Climate Change, UNFCCC, United Nations Framework Convention on Climate Change. That means a convention given, a framework of convention given by United Nations to manage or mitigate climate change. Kyoto Protocol is one such thing. So the agreements was countries took targets in reducing emission reduction, defining what greenhouse gases are, what are the gases that we should consider as greenhouse gases, and coming into some agreements on how do we go about dealing with it, how concessions can be applied. There were already countries which were very heavily industrialized countries, even in 1997, heavily industrialized countries. If you apply the emission norms to those countries, they will have to stop production. They will have to stop their industrialization. So this climate change is being looked at as the responsibility of ensuring the global climate management. Global climate remains in a particular level, at a particular level, or degradation happens below a certain level. So these kind of conventions also discuss on concessions. What concessions? You are a non-industrialized country. I am already an industrialized country. So if I have to agree to the emission norms, then I will have to cut down on my industrial production. I will, however, work on reducing emissions from the existing industries, but that will not still be kept enough to match the emission standards, emission cutoffs. So what do? I will give emission credits to another country, which is a non-industrialized country. I tell them, you don't become industrialized, I'll give you money. So that together, my emissions and your emissions put together is in a balance. So fair, no? Right? United Nations is a bogus cartel of a few countries which will try and manipulate every other country for their own benefit. Until India and one of the African nations become a part of core decision making in the United Nations, I consider it as a self-serving bogus entity which serves the interests of only a few countries in the Western world. How can you keep two continents out of decision making for the world? India and Africa is not represented. And in Asia, India is a very prominent country. 1/7 of the world's population reside in India. So, all right. Anyways, greenhouse gases coverage, flexibility mechanisms, emission trading, all this used to happen in these conventions. Which of the following represent the maximum number of species among global biodiversity? Algae, lichens, fungi, mosses and ferns. Which one? Fungi represents the largest groups on biodiversity. 1.5 million species apparently. Algae, lichens, mosses, ferns, these are also abundant, but not as abundant as fungi among the given four. Match the name of the animals in column 1 with one characteristic and the phylum to which it belongs. Petromyzon, ectoparasite, phylum or class Cyclostomata. Petromyzon is also referred to as lamprey. It is an ectoparasite, attaches to fishes and feed on their blood. They belong to Cyclostomata only, class like Cyclostomata. That is correct. Let us look at the others. Ichthy, terrestrial reptilia. Ichthy is not terrestrial, it is aquatic and it belongs to fishes, pisces, not reptiles. Limulus is the crab, king crab or horseshoe crab, whatever the crab that we find. Body covered by chitinous exoskeleton. Is it true? Characteristic? Crab has exoskeleton made up of chitin. It belongs to Pisces is the statement given there. It doesn't belong to Pisces. Crab belongs to arthropods. Adamsia or sea anemone is radially symmetrical and it belongs to not Porifera, Coelenterata. Okay, that's all. Which of the following are correctly matched with respect to their taxonomic classification? Same combinations you can remember. Mix and match of these things can come. Flying fish, cuttlefish, silverfish. Flying fish and cuttlefish are fishes, but silverfish is insect. Silverfish is an insect. So statement 1 is wrong. Option 1 is wrong. Centipede, millipede, spider, scorpion. Centipede, millipede, spider are insects, but scorpion is arthropod. Scorpion is arthropod. Housefly, butterfly, tsetsefly, silverfish. Housefly, butterfly, tsetsefly, silverfish are all insects only. Statement 3 is right. Spiny anteater, sea urchin, sea cucumber, echinodermata. Spiny anteater is a mammal. Spiny anteater is a mammal. Is it not an oviparous mammal? It is an egg-laying mammal. Spiny anteater is an egg-laying mammal. Okay, that is the example for egg-laying mammal. Sea urchin and sea cucumber are echinoderms. What is the disease that tsetsefly can transmit? Sleeping sickness. What is the parasite in sleeping sickness? Trypanosoma. So it is also called trypanosomiasis. Trypanosoma will parasitize. It is a parasite of the nervous system, causing drowsiness and sleeping sickness. Your daytime sleepiness is not because of trypanosoma. Okay, so don't relate your sleepiness to sleeping sickness. Sleepiness or the sleeping sickness of students is because of the assault of teachers and not parasite. All right. Which group of animals belong to the same phylum? Malarial parasite, amoeba, mosquito. Malarial parasite belongs to Protista. Protista. What is the parasite? Plasmodium. Plasmodium is a protistan. Amoeba is a protozoa. Mosquito is arthropoda. They all don't belong to the same phylum. Earthworm, pinworm, tapeworm. Earthworm is what? Annelid. Earthworm is an annelida. Pinworm is nematode. Pinworm, roundworm, all of these are nematodes. Tapeworm is platyhelminthes. Tapeworm is platyhelminthes. Again, all of them don't belong to the same class or the same phylum. Prawn, scorpion, locusta. Locusta is locust. Prawn, scorpion, scorpion and locusts all belong are all arthropods. Sponge, sea anemone, starfish. Sponge is porifera. Sea anemone is Cnidaria. Starfish, Echinodermata, different phylum. So prawn, scorpion and locusts belong to the same phylum, which is Arthropoda. One of the representatives of phylum Arthropoda is cuttlefish, silverfish, pufferfish, flying fish. Silverfish is an insect. Insecta is a class of phylum Arthropoda. Cuttlefish is a mollusk. Pufferfish and flying fish are fishes, actual fishes, which belong to Pisces. Pufferfish. That is the one which puffs, no? Becomes a ball of air. H zone in the skeletal muscle fiber is due to the absence of myofibrils in the central portion of the A band, central gap between myosin filaments in the A band, central gap between actin filaments extending through myosin filaments in the A band, extension of myosin filaments in the central portion of the A band. See, there are different zones. Just quickly, I will write the sarcomere. You just recollect the zones. What is the boundary of the sarcomere? Z lines. Then you have thin filaments extending across the Z lines. Thin filaments extending across the Z lines. Correct? Then you have thick filaments in between the Z lines, between the thin filaments. Thick filaments in between the thin filaments. This Z line is a fibrous tissue. Z line is a fibrous tissue holding the thin filaments in place. There is a thin fibrous tissue in the middle of this segment also holding the thick filaments in their position. What is this? M line. Now this zone is H zone. This band is or here up to this. This band is A band. This band is now again till here. This band is I band. Isotropic band and anisotropic band. Isotropic band is, it appears similar. The entire stretch of the band, it appears similar. See, isotropic band is across sarcomere. It is the zone which is having only the thin filaments on either side of the sarcomere, or either side of the Z line. That is the I band. A band is within each of the sarcomeres. That is the zone which is overlapping between the thick filament and thin filament, including the H zone. H zone is also a part of the A band. Band includes that H zone also. H zone is this zone where there is no where there is no overlap between the thick and thin filaments in the center. Only the thick filament is present. That is the H zone. H zone. So what shortens when the sarcomere contracts? A band, does it reduce in size? A band doesn't reduce in size. I band doesn't reduce in size. H zone is the one that becomes narrow. Because the thick filaments are the thin filaments are pulled closer to each other by the thick filament. Alright. So look at the options now. H zone is due to the central gap between actin filaments extending through the myosin filaments in the A band. That is the H zone. Is H zone in the A band lighter or darker in its appearance? H zone should be darker. Why? It has only the thick filament contributing to its visibility. In the other part of the A band, there is thick filament and thin filament also. So which one will be darker? The A band outside of the H zone will be darker. H zone will be the lighter zone within the A band. H zone will be the lighter zone within the A band because there is only thick filaments there. In the other portion, on either side of the H band or H zone, in the A band, you have thick and thin filaments overlapping with each other, increasing its, what do we call it? Making it more darker. All right. Okay. Echogenicity, you can say. All right. What external changes are visible after the last molt of a cockroach nymph? Do you want to go through it? Cockroach is not there in our syllabus anymore. Huh? It's there? Cockroach is not there, no? It is frog. Frog is a part of our syllabus now. Cockroach has been removed. No? Cockroach is there? Okay. What external changes are visible after the last molt of a cockroach nymph? Mandibles become harder, anal cerci develop, both forewings and hindwings develop. Last molting. So what is the last thing that will happen in the cockroach molting? Wing development. Labium develops. Mandibles, anal cerci, labium all develop in the earlier stages of molting. The last thing that happens in the molting is, last thing that happens when the nymph becomes a adult is development of wings. Which wing is more thicker in cockroach? Forewing or hindwing? Forewings are more darker and harder in texture. Okay. Golgi complex plays a major role in trapping light and transforming it into chemical energy. Which organelle is responsible for that, trapping light and transforming it into chemical energy? Chloroplast. In digesting proteins and carbohydrates, if at all we have to look at organelles doing this, which organelle will do that? Lysosome or the vacuole, food vacuoles. Food vacuole and lysosome fuses to form that, do that. As energy transferring organelles, which organelle can transfer energy? Mitochondria, transformation and transfer of energy. Mitochondria can do that. In post-translational modification of proteins and glycosylation of lipids, Golgi complex does that. Golgi complex is a modification center. It is an alteration, modification, restructuring, packaging center. It can modify chemicals, it can alter their chemical structure, it can restructure, bring about combinations. All of that mix and match happens in the Golgi complex. Very powerful. Post-translational modification of proteins, glycosylation and phosphorylation of lipids. Which one of the following organelles in the figure correctly matches with its function? Which organelles are given there? What organelles do you see? What organelles do you see in the diagram? Huh? What all do you see? You see nucleus. You see nucleus. You see rough endoplasmic reticulum. Do you also see smooth endoplasmic reticulum? Do we find Golgi complex? No. We see rough endoplasmic reticulum, smooth endoplasmic reticulum and nucleus. Okay. Rough endoplasmic reticulum, formation of glycoproteins. Is it right? Which organelle will produce glycoprotein? Golgi. Golgi apparatus, protein synthesis. Is it right? Golgi apparatus does not participate in protein synthesis, and also Golgi apparatus is not there in the diagram. Golgi apparatus, formation of glycolipids. Formation of glycolipids is correct, but Golgi apparatus is not there in the diagram. So the question is related to the diagram. Rough endoplasmic reticulum, protein synthesis. That is right. Golgi apparatus is responsible for modifying, sorting of chemicals, packaging the chemicals and delivering those chemicals in the form of vesicles also. Macromolecule chitin is nitrogen containing polysaccharide, phosphorus containing polysaccharide, sulfur containing polysaccharide, simple polysaccharide. Chitin is a combination of two chemicals, two polysaccharides. What are they? N-acetyl muramic acid and N-acetyl glucosamine. N-acetyl glucosamine and N-acetyl muramic acid. And chitin is a nitrogenous polysaccharide, nitrogenous heteropolysaccharide, nitrogen containing heteropolysaccharide. Where do you find it? Where do you find chitin? Fungal cell wall. The cell wall of fungi has chitin, and also the exoskeleton of arthropods. Exoskeleton of arthropods also contain chitin. Essential chemical components of many coenzymes are what? Vitamins. Vitamins are, the importance of vitamins is that they play the role of a coenzyme. Coenzyme, prosthetic group, what is the difference? Prosthetic group is tightly bound. Coenzymes are separable. You can separate them. They are not bound to it inseparably. A stage in cell division is shown in the figure. Select the answer which gives correct identification of the stage with its characteristics. What do you notice? Look at the diagram and tell me what all do you see. Centrioles are present. Spindle fibers are present. Then, do you see a nucleus? Do you see nuclei on both ends? Two nuclei are present. Nuclear membrane is clearly visible. Nuclear membrane is clearly visible. Do you see chromosomes? Do you see chromosomes in the nucleus? You don't see chromosomes in the nucleus. That means the chromosomes are all diffused. Decondensation of chromosomes has happened, and they are in the form of a chromatin mass. They are in the form of chromatin mass. So whenever we look at a stage, you should look at the stage at the end of it. If you say prophase, you will talk about what do you see at the end of prophase. When you say metaphase, you will talk about what you see at the end of metaphase. Same with anaphase and telophase. Now here, if it was prophase, what should you have seen? Will you see centrioles at the end of prophase? Do you see centrioles at the end of prophase? Yes. Do you see spindle fibers at the end of prophase? You do see spindle fibers also. Will there be nuclear membrane at the end of prophase? No nuclear membrane. Can you see chromosomes at the end of prophase? Yes. But chromosomes are not aligned. At the end of metaphase, centrioles are there. Chromosomes are lined up at the equator. Spindle fibers are attached to the centromere regions of the chromosomes. That is the end of metaphase. At the end of anaphase, chromosomes are at the two poles of the cell. Spindle fibers are still there, centriole is still there. At the end of telophase, chromosomes decondense, nuclear envelope forms, centrioles and spindle fibers disappear. Centrioles and spindle fibers disappear. Here this diagram does not correspond to the end of telophase also. It is not end of prophase because you don't see chromosomes. Nuclear membrane is still there. Alright. So look at the options. Option 1, telophase. So this is not a representation of any particular end stage. Any end of any particular stage. But look at the options and try to come to a conclusion. Telophase, nuclear envelope reforms. Maltose has to be further digested before it is absorbed. Maltose is broken down into what before absorption? Maltose is broken down into two molecules of glucose. Glucose and glucose. So, glycine, glucose, absorption in small intestine via active absorption can be the right choice. A pregnant female delivers a baby who suffers from stunted growth, mental retardation, low IQ and abnormal skin. This is the result of. If a baby is born with these things, there can be chromosomal abnormalities or there can be metabolic abnormalities. If it is metabolic abnormalities, something that should have happened in the baby has not happened. We will look at the options and try to figure out. Deficiency of iodine in diet of the mother. Is it possible that iodine deficient diet of the mother can lead to these symptoms, can lead to this manifestation? Stunted growth, mental retardation, low IQ and abnormal skin. How? Thyroid development of the fetus would not have happened properly because of lack of iodine. Thyroid hormone production in the fetus would have been low. And all these signs and symptoms are characteristics of cretinism, thyroid hormone deficiency. Okay. Low secretion of growth hormone. Why not this option? Why not low secretion of growth hormone? Stunted growth can be seen there also, no? Growth hormone deficiency does not lead to mental retardation. Both growth hormone and thyroxine or thyroid hormone contributes to growth. But thyroid hormone also contributes to mental development. Absence of growth hormone or deficiency of growth hormone will not lead to mental retardation. Mental development will be adequate only, provided thyroxine is normal. Number 3, cancer of the thyroid gland. Is it possible? Cancer of the thyroid gland of the mother. Can that lead to baby developing hypothyroid symptoms? Possible. Why not? Possible. Cancer can be over-secreting cancers or under-secreting cancers. So cancer can lead to overproduction of thyroid hormone also in the mother. So you cannot conclusively say that thyroid problems in the mother will lead to thyroid problems in the baby. Baby's thyroid development should happen. The changes in the baby is seen because the baby's thyroid gland is not developed. That is not related to the mother's thyroid status. It is related to the supply of iodine and the other essential nutrients for the development of the thyroid gland. If the baby is continuing to stay till 28 weeks of pregnancy in the uterus, the baby will develop the thyroid gland. Development of thyroid gland will lead to production of thyroxine hormone. For thyroxine hormone to be produced, iodinization should happen of the thyroxine amino acid. So iodine is required. In the absence of iodine, even if the thyroid gland development of the baby is proper, thyroid hormone will not be produced in the baby, which will lead to the symptoms. So mother's thyroid status, whether the mother is hypothyroid, hyperthyroid, mother has thyroid cancer, all of that will not impact the baby as much as iodine deficiency would. Over-secretion of pars distalis. Pars distalis is what? What is pars distalis? Anterior pituitary? Anterior pituitary? What do you call posterior pituitary? Pars nervosa. Anterior pituitary over-secretion of anterior pituitary, can that lead to thyroid deficiency? Over-secretion of anterior pituitary, assuming that the baby's anterior pituitary is over-secreting, it will lead to hyperthyroidism. It will not lead to hypothyroidism. TSH will be produced more, thyroid gland is stimulated more. If the iodine deficiency is not there, it will lead to hyperthyroidism, not hypo. So the right choice is iodine deficient diet. The mother's diet not having enough iodine will lead to baby's thyroid development deficiency more than any of the other causes given. The figure shows a diagrammatic view of the human respiratory system with labels A, B, C and D. Select the option which gives correct identification and main function and or characteristics. Just look at the labels and tell me what each one is. A is what? Trachea. B is pleura. C is alveoli. D is diaphragm. Let's look at the options. A, trachea, long tube supported by complete cartilaginous rings for conducting inspired air. True or false? What is wrong about the statement? It is not complete ring, it is C-shaped ring, incomplete cartilaginous ring. So statement A is wrong. Pleural membrane surrounds ribs on both sides to provide cushion against rubbing. True or false? What is wrong about the statement? It is a wrong statement. What is wrong? It does not cover the ribs on both sides. It covers the outer surface of the lungs and the inner surface of the thoracic cavity. It is not covering the ribs on both sides. Alright. Alveoli, thin-walled vascular bag-like structures for exchange of gases. That is true. D, lower end of lungs. Diaphragm pulls it down during inspiration. It is diaphragm only. It is not the lower end of lungs. D, the label is wrong in option D. So the correct answer is 3C, alveoli, thin-walled vascular bags-like structures for exchange of gases. Figure shows a schematic plan of blood circulation in humans with label A to D. Identify the label and give its functions. A, B, C, D. Okay. So double circulation is given. You just identify A, B, C, D. What is A? Pulmonary veins. B is aorta and its branches. C, inferior vena cava. D is superior vena cava. All right. No, D is not superior vena cava. D can be pulmonary artery also. But let us look at, see, it is not necessary what is given on top is pulmonary and what is given at the bottom is systemic circulation. We look at the options. A, pulmonary vein takes impure blood from body parts, PO2 60 millimeter mercury. Wrong. Pulmonary vein takes oxygenated blood from lungs to the left atrium. Partial pressure of oxygen, PO2 will be. Do you remember the partial pressures that I told you? 140, 40, 46. These are the four numbers you remember. 140 for oxygen, 40, 46 for carbon dioxide. If it is oxygenated blood, 100, deoxygenated blood, 40. Carbon dioxide is more, 46, carbon dioxide is less, 40. Carbon dioxide will be more in what blood? In deoxygenated blood. If the sample of blood is collected from a person's body and the sample of blood showed partial pressure of oxygen to be 40 millimeter mercury, what do you expect the partial pressure of carbon dioxide to be in this blood? 40? 45 millimeter mercury. 45, 46, around that, just remember that. Let's say 45 millimeter mercury is the partial pressure of carbon dioxide. If the sample of blood collected from the person's body shows partial pressure of oxygen to be 90, what will be the partial pressure of carbon dioxide? 40. We are not telling 100 here. The cells in that sample also would have used oxygen. So 40. Okay, B, pulmonary artery takes blood from heart to lungs, PO2 90 millimeter mercury. Pulmonary artery, it can be. B can be pulmonary artery, but takes from heart to lungs. Okay, possible. Let us give it also. PO2. PO2 given is 90 millimeter mercury. Wrong. Pulmonary artery carries deoxygenated blood whose partial pressure of oxygen will be around 40. C, vena cava takes blood from body parts to right auricle. PCO2 is 45 millimeter mercury. Can be right. Can be right. D, dorsal aorta takes blood from heart to body parts. Partial pressure of oxygen is 95 millimeter mercury. That also can be right. Now, we have to make a judgment call here. We have to make a judgment call. Statements are correct. C and D, if you don't look at the label and only look at the structure, vena cava carries blood from body parts to right auricle, PCO2 45 millimeter mercury. Is it correct? Don't look at the label. Just vena cava, what it does and what is the PCO2. It is correct. Dorsal aorta takes blood from heart to body parts, PO2 95 millimeter mercury. Correct? Statement wise, aorta carries blood from heart to all the body parts. That is also correct. Which one do we choose now? Between C and D. C can be vena cava. Can D be? If D is dorsal aorta, then A should be. If D is dorsal aorta, A should be vena cava. C cannot be vena cava. If C is vena cava, D cannot be dorsal aorta. D has to be pulmonary artery. D has to be pulmonary artery. What do we do now? Now we have to look at the technicality of the diagram. Look at the technicality of the diagram. Which circulation is longer? Systemic or pulmonary circulation? Systemic circulation is longer in its span. Now in this, D is a part of, A and D are a part of which circulation in that sense? A and D are a part of pulmonary circulation. B and C should be part of systemic circulation. In that sense, which one should be the right choice? C should be the right choice. Clear? So, what is given has to be analyzed and you have to come to a conclusion. The diagram given here is the standard ECG of a normal person. P wave represents what? P wave, P, QRS and T. Let me see what all you remember of ECG. P wave represents, what all does P wave represent, you tell me. P wave represents in the electrical conducting system, what does it represent? Firing of the SA node. In the electrical conducting system, P wave represents firing of the SA node. In the cardiac cycle, what does P wave represent? In cardiac cycle, what does it represent? Atrial systole. In the cardiac cycle, it represents atrial systole. Alright. So P wave represents both. And in terms of the action of the cardiac muscles, what does it represent? Contraction of atria. What will be the ventricle in that stage when P wave is formed? Ventricles will be in a state of relaxation or diastole. Okay. QRS complex associated with firing of AV node, contraction of ventricles, relaxation of atria, ventricular systole. T wave, ventricular repolarization. Okay. Ventricular repolarization, and it corresponds to what phase of the cardiac cycle? T wave corresponds to joint diastole. Now let us look at the options. P wave represents what? Contraction of both the atria. True or false? True. Initiation of the ventricular contraction. False. Beginning of the systole. True, but partially true. Systole of what? Atrial systole or ventricular systole? It is incomplete. Option 3 is incomplete. End of systole? False. It is corresponding to atrial systole. So contraction of both atria is the right choice. The figure shows human urinary system with structures labeled A to D. Select the option which correctly identifies them and gives them characteristics and or functions. What is A in the diagram? We will recognize them, then we will look at the options. A is, what is A? Adrenal gland. B, pelvis, renal pelvis. C, if you look at it, it is pointing to renal pyramids. D, renal cortex. Now look at the choices, options. A is adrenal gland, correct. Located at the anterior part of the kidney, secretes catecholamines which stimulate glycogen breakdown. Catecholamines are what? Adrenaline, noradrenaline, or epinephrine, norepinephrine. Same. Is it right? A, option 1. It is correct. Located in the anterior part of the kidney, secretes catecholamines stimulating glycogen breakdown. By stimulating glycogen breakdown, what are these catecholamines trying to do? They are trying to give you additional supply of glucose. Why? To overcome stressful situations. Catecholamines or adrenaline, noradrenaline are stress hormones. They are released in response to stress. Why do people put on weight when they are in excess stress? Can anybody explain at this stage with this? Why do you put on weight when you are experiencing high levels of stress? What is the relationship between stress and weight gain? When you are hungry, what hormone is produced in the body? Glucagon is produced. What will glucagon do? Glucagon will cause breakdown of glycogen and make glucose available in your blood. To what level? Glucagon can bring the glucose level to about 100 to 110 milligram per deciliter. So you don't feel hungry, but you don't have excess glucose in blood to push it into storage. Alright. Glucagon and insulin function in contrast. So when glucagon is acting, insulin will not act. You understand? When glucagon is active, insulin does not act. It allows glucagon to function and maintain the blood glucose level. When insulin is acting, that when will insulin get activated? When there is high glucose level in the blood. High glucose level, insulin gets activated. What will insulin do? It will quickly try to push glucose into compartments for storage and utilization. Cells will utilize, and whatever is in excess, higher the levels of insulin coming into your blood, higher will be glucose getting pushed into storage. Because the utilization by cells is constant, it is more or less going on at a continuous pace. If more insulin is available, it will try to push it into compartments. You understand? Remember this one. Insulin will stimulate liver to convert glucose into glycogen. Insulin will stimulate adipose tissue to convert glucose into fat. Point number one. But when glucose level is high, glucagon is not adding to more glucose. It will stop. When glucagon is breaking down glycogen to bring in glucose, insulin production will not happen. So insulin is keeping quiet, letting the glucose to be utilized by cells for their metabolism. Catecholamines are independent. When you are in a stressful situation, adrenaline, noradrenaline is released in the blood, which will make sure that glycogen is broken down and more glucose is pushed into bloodstream. More and more influence of adrenaline, noradrenaline, your blood is constantly having high levels of glucose. When blood is having high levels of glucose, what will it do to insulin? What will happen to insulin production? Insulin production goes up. Insulin production goes up. What will insulin do? It will push this glucose into storage. When you push this glucose into storage, you are breaking down glycogen on one side. Insulin is pushing that glycogen back into storage, stimulating conversion of that into fat. Reducing glycogen levels will trigger the mechanism of restoring the glycogen in the body. So that will translate into increased hunger. It will stimulate the hypothalamus. Hypothalamus will make you feel more hungry. And you eat more. When you eat more, blood glucose level further increases. Insulin will further push it into storage. And because of stress, continuously these fellows, catecholamines, adrenaline, noradrenaline, are breaking down glycogen. So glycogen stores are continuously being depleted by catecholamines, making glucose enter bloodstream. Glycogen levels go down, triggers a mechanism of restoring that, because that is emergency supply of energy. Increasing hunger. You eat more, glucose level rises. Add to that breakdown of glycogen, adding to more glucose, more insulin production, push it into storage, more fat production. See that? Why it is important to do yoga. Alright? Yoga is the best form of physical activity and mental conditioning available for human beings, which will correct a lot of things. And high level of glucose in the blood increases inflammation. Body inflammation increases with high levels of glucose. Increases infection possibilities because high levels of glucose is conducive for microorganisms to grow. Increases load on the kidney because you have to constantly reabsorb this high level of glucose. More it gets filtered out, you have to take back. Increases load on the kidney. Increases fluid retention in blood. Increasing blood volume. Increasing blood pressure. Alright, you see that? Just stress. If you can take care of three things in your life on a day-to-day basis, how you eat your food. The second part is your activities. 1 hour of yoga every day, 1 hour of outdoor sports every day. Number 3, rest. Compulsorily 6 hours of sleep in the night, uninterrupted. Short intervals of sleep during the daytime. Practicing to take power naps. 15 minutes of power nap will restore energy to do 3 hours of work. But you have to practice doing power nap. When I initially tell students to practice doing power nap, taking power naps, many of you also complained about that. Sir, I cannot sleep for 10 minutes. If I sleep, when I wake up it will be 2 hours. I cannot sleep for 10 minutes. But if you practice in a controlled condition, you are forced to wake up at 10 minutes. No? You have to come to the class, otherwise there will be some consequence of that. So you are forced to do that. When you are forced to comply to something, whether out of force or out of will or out of understanding how it impacts, you do something, it becomes a habit. When it becomes a habit, you learn to sleep for 10 to 15 minutes. It gives you so much power. So you have to take care of your food, your activities and rest. Did you understand stress? The effect of stress is permanent. Adrenaline and noradrenaline is not only acting on glycogen, breaking down glycogen and increasing blood glucose level, triggering insulin. Insulin will go and push it into storage, putting you in a perpetual cycle of hunger, eat more. Even when you are eating more, these fellows are breaking down glycogen and bringing it into glucose, into bloodstream. Stress, pushing it into storage. A lot of people will come across. Okay. I have put on a lot of weight because of stress. I don't eat much. I eat very limited food. I don't have too much of eating craving. I have a balanced lifestyle. But because of too much work, doing too many things, stress catches up. And even without eating much, you will start putting on weight. Whatever you eat, your body, see there is expenditure mechanism and storage mechanism, savings mechanism in the body. It is like economics. If your expenditure mechanism is very active, what you eat will be utilized and excess is not bothered much. The body will not bother much about excess. It will store minimally because there is always availability. But if your savings mechanism in the body is activated, every bit of energy available will be converted into storage. Very efficient conversion of excess energy into storage form, which is fat. That will happen. So stress will trigger evolutionary survival instincts in the body. There is survival instinct operating at the brain level. There is survival instinct operating at every cell level. Cellular level survival instincts are there. Every cell will get triggered to survive. And the best way to do that is store everything. So handling stress is extremely important. Understand? Adrenal gland. So we went into catecholamines, stimulating glycogen breakdown, all of that. B, pelvis, broad funnel-shaped space. Statement 1 is correct. Pelvis is a broad funnel-shaped space inner to hilum directly connected to loops of Henle. Is it right? Wrong. Pelvis is not directly connected to loops of Henle. It is the end of that entire transportation channel. Loops of Henle are connected to collecting ducts. Medulla, C, inner zone of kidney and contains complete nephrons. First of all, C is not medulla, C is reference to pyramids. And medulla does not contain the entire nephrons. Medulla contains the collecting PCT, DCT loops and these things. The capsule, the renal capsule or the renal corpuscle is in the cortex of all nephrons. D, cortex, outer part of kidney and does not contain any part of nephrons. Cortex is correct. Outer part of kidney is correct. But does not contain any part of nephron is wrong. It contains parts of nephron. So statement 1 is right. Don't you think as a part, whatever I am sharing with you right now, this is irrespective of what you choose to do for your career, for your life, for your money, lifestyle, all of that. This is all knowledge or understanding of yourself which will help you manage your life better. What do we have in our school textbooks? All nonsense. We should have things, it's knowledge only, no? We should have things which will make people understand themselves, understand their bodies functioning, implications of that. Stress is so important. Why we have to focus so much on managing stress? And it is not once a month workshop or once a month some session I attend. I go to the psychologist and take counseling. You have to manage it every day. You have to have mechanisms of managing stress on a day-to-day basis. You need to have stress relieving mechanisms and mechanisms internalized which will prevent you from accumulating stress or influence of stress. Yoga. No, it's not a joke. You see, the only resolution accepted by all countries of the world, June 21st is celebrated as International Day of Yoga. All the countries of the world endorsed yoga as something to be promoted and propagated across the world for the welfare of humanity. There is nothing else that has been passed with such high majority of countries voting in favor of that resolution. Alright, only the resolution to make June 21 as the International Day of Yoga. India's gift to the world. We are the masters of that. India is the master of human management. No other country has got this vast knowledge of how to handle human beings, our own selves. Alright, psychology appears like child's play in front of the ocean of technology we have in our country. Alright, so human management, we are the masters. Technology, we are catching up. External technology. Oh my God. Select the correct statement with respect to locomotion in humans. A decreased level of progesterone causes osteoporosis in old people. Correct the statement. A decreased level of estrogen causes osteoporosis in old people. Estrogen and demineralization of bone are related. Can we talk about estrogen and demineralization or mineralization of bone for all human beings? It is predominantly in women. Estrogen has an osteoprotective effect. That means it will prevent demineralization from bone. Absence of estrogen removes that protection. That means the bones are subject to the demineralizing women, more common in women. So, post-menopausal women need to monitor their bone density. They need to monitor their bone density, and if required, hormone replacement therapy should be given. That is, low doses of estrogen given continuously for post-menopausal women. Important. Can we do that for men also? Elderly men can be given estrogen? We should not do that. Testosterone has a mineralizing effect on the bone. Testosterone promotes bone mineralization. It helps the bone become stronger. So testosterone production continues for lifetime. So we don't need to have estrogen protection for bones of men. Okay, don't start using it. Estrogen has other effects also. All right. Accumulation of uric acid crystals in joints causes their inflammation. True or false statement? Statement is true. Uric acid accumulation in joints is referred to by a name, a disease, which is gout. Gout. Inflammation of the joints is referred to by a term, arthritis. Arthritis. So inflammation caused due to deposition of uric acid crystals is called gouty arthritis. Gouty arthritis, rheumatoid arthritis, osteoarthritis, three types of arthritis you should consider. Gouty arthritis, uric acid crystals. Rheumatoid arthritis, autoimmune disease, where the joint capsule or the joint fluid is or the joint is considered as a foreign body and the immune system starts attacking that. That is rheumatoid arthritis. What is osteoarthritis? Any other cause, not neither deposition or age-related inflammation of the joints, can be injury or rubbing of the bones. Synovial fluid might become lesser. Any of those conditions causing inflammation. Vertebral column has 10 thoracic vertebrae. True or false? How many thoracic vertebrae are present? 12. 7 cervical, 12 thoracic, 5 lumbar, 5 sacral fused into 1, coccygeal, 4 coccygeal bones fused into 1. This is the number of bones. The joint between adjacent vertebrae is a fibrous joint. It is a cartilaginous joint. Adjacent vertebrae, intervertebral joints have limited movement, but they still have movement. It is not a fibrous joint. Fibrous joint is immobile joint, doesn't move. Characteristics of and an example of a synovial joint in humans is fluid cartilage between two bones, limited movements. Knee joint. Is it right? Knee joint. What type of joint is knee joint? It is a synovial joint. The example, it's all of them are synovial joints only here. Knee joint is a hinge joint. One plane movement. Is it fluid cartilage that is present between the bones in knee joint? It is synovial fluid. That fluid is not cartilage there. Alright? It's a lubricating fluid. Fluid filled between two joints provide cushion. Skull bones. Is it right? Skull bones. The joint between skull bones is fibrous joint. There is no fluid. Fluid filled synovial cavity between two bones. Joint between atlas and axis. Atlas and axis is what joint? Pivot joint. Alright? That is movement along the axis of one bone. That is pivot. So that is correct. Lymph filled between two bones, limited movement. Gliding joint between carpals. Gliding joint is correct. Between carpals you have gliding joints. But is it lymph filled between the two bones? It is not lymph. It is synovial fluid. Alright. Tell me the joints, different types of joints. Ball and socket. Ball and socket. The most versatile ball and socket joint is which one? Which one is the most versatile ball and socket joint? Shoulder. Shoulder has the highest range of movements. Alright. Then hip joint is also ball and socket. Then hinge. Hinge is elbow and knee. Pivot joint, atlas and axis. Atlas axis and radio-ulnar joint. Radius and ulna. Radius and ulna, this joint. Here. You can do this, no? Radius and ulna slides over each other. That is radio-ulnar joint. Alright, then saddle joint. Metacarpophalangeal joint. Metacarpal and phalanx. Two plane movement. Saddle joint. This one and you will do this. Two plane movement is saddle joint. Then we have plane joint or gliding joint between the carpal bones, carpal bones. And wrist joint is ellipsoid joint. Ellipsoid joint, two plane movement again. Alright, flexion extension is possible. Lateral movement is also possible. Abduction, adduction. Abduction is moving away from the midline. See, this is the joint, this is the wrist joint. Abduction is moving away from the midline. Adduction is moving towards the midline. Which range is much more in wrist joint, abduction or adduction? Abduction is more. It's of a higher angle than adduction. So shoulder joint, abduction is this, adduction is this. Alright, like that. Flexion extension is extending the joint and flexing the joint. Alright. One last one we'll do, 155. A diagram showing axon, terminal and synapse is given. Identify correctly at least two of A to D. So it is 2, 2. We'll identify A to D, then we will look at it. What is A? A is the receptor. It is on the post-synaptic membrane. B, look at the diagram. B is synaptic cleft. It is not pointing to the neurotransmitter. It is pointing to the space, synaptic cleft. C is neurotransmitter or the synaptic vesicle. It is pointing to the neurotransmitter there inside. If it is synaptic vesicle also, we can take it. Synaptic vesicle with neurotransmitters. D is neurotransmitter. D is neurotransmitter. All right. Statement 1, option 1, A is receptor. Correct. C is synaptic vesicle. Correct. Let us see if there are any better options. Statement 2, B is synaptic connection. There is nothing called synaptic connection there. D is potassium. No. Which ion contributes to fusion of the synaptic vesicles to the presynaptic membrane and release? Calcium ion contributes to release of release of neurotransmitters into the synaptic cleft. But is D calcium? Is D calcium? D is not calcium. D is neurotransmitter. So 2 is wrong. 3, A is neurotransmitter, wrong. C is neurotransmitter. Okay, we can take that. But D is mentioned as calcium. That is wrong. Alright, so statement 1 is correct. Option 1 is correct. Rest of it we will continue. Alright, we will continue in the next class.